Welcome and thank you for standing by. At this time, all participants are on a listen-only mode until the question and answer session of today's conference. At that time, to ask a question from the phone lines, please press star 1 and record your name when prompted. This conference is being recorded. If you have any objections, please disconnect at this time. I would now like to turn the call over to your host, Irene Ahir. You may begin.
Thank you. Hello, I am Irene Ahir of CDRH's Office of Communication and Education. Welcome to the FDA's 20th in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Timothy Stencil, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, and Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to today's discussion. Please remember that we are not able to respond to questions about specific submissions that might be under review. Now, I give you Timothy.
Thank you, Irene, and hello again, everyone, and thank you for joining us, and we greatly appreciate everything you're doing to help out in this pandemic. We passed a new milestone recently. We have authorized now over 200 tests in the UA pathway, and thanks to many of you for that accomplishment. Without developers like yourselves doing this, we could not have achieved that goal. We are going to have many, many more. We have hundreds of EUA applications in-house, and the vast majority of these have come through the notification pathway, so that they are able to, if they do that, if that's allowed to them, you are all able to go out and market your test prior to EUA authorization. It is the honor system. We expect that the validations have been done well, the performance is acceptable, and we thank you for that. We allowed this pathway in this pandemic because of the absolute huge need to provide testing in abundance as soon as possible. And so please do bear with us as we go through these applications for EUA authorization. We obviously have to prioritize these so that we make the best decisions possible in all cases. So everyone's EUA is important, and we will get to them. Please work closely with the contact that we've provided. Again, for EUAs, not pre-EUAs, for EUAs, directed the office, make sure that every EUA applicant has a contact within 2 weeks of submission, hopefully less time now. And that's important, that you have a contact, that you can get at least weekly updates from. And those that do require an EUA prior to launch, they obviously get triaged. For those new submissions, new technologies that require an EUA, they do get higher priority because they are awaiting EUA authorization to be able to get to the market versus those who come through the notification pathway. The second update has to do with pooling. Of course, there's a greater need for tests than available tests, and turnaround times at labs have increased. One of the potential solutions to this is that we find a way to pool. Pooling, in all cases, appears to decrease the percent positive rate. That is, low positives are going to be missed. and that's just a given if you start pooling. We want to limit those misses to an acceptable level, and our updated templates call for a PPA relative to single tests on the platform of 85%. You can pool up to as many as you want as long as you hit that number. We are asking for an even distribution of levels of positivity, so a good range of low positives and high positives, up to high positives, so that there's not an overabundance of either. An overabundance of low positives, if it doesn't match your distribution in your lab, which is the ideal thing, is to match the distribution your lab sees, then that it's not as helpful. So we'll work closely with you to make sure that that distribution is good. The pathway for pooling additions, both for kit developers as well as labs to alter kits and or alter their own LDTs, is through the notification pathway. So validate whatever pool scheme you want to use, make sure the performance is good. On the honor system, you can notify us and submit your data within 15 days, all the while, 15 business days, all the while continuing to test pools unless you hear any concerns from us. And we'll get to those applications as soon as possible. They are a priority now so that we understand the variables that go into developing a pool scheme. There are multiple schemes. We have seen tremendous variability between labs that have tried to validate pooling, even with the same kit. So, for example, somebody might fail the bar at a lower sample pool where another lab seems to pass. And so we are gaining more and more information about why that might be. It seems to be driven by two different things, at least. But these are probably the major drivers of pooling success, and that is the LOD of the assay. Obviously, a more sensitive assay will be less likely to miss the average low positive than a test that is pooled that has not as great a sensitivity. And then second is the distribution of low positives in a given lab's menu and experience. So it's important to understand historically what your low positive rate is. We like to see CTs historically for the labs to help them, guide them to the right pool scheme that will give them great performance with pooling, and balance the need to increase throughput with getting acceptable performance and not missing a lot of positives. And then finally, I wanted to mention that recently we did authorize the first semi-quant serology test. In fact, there were two of them that we authorized. Semi-quant, quant, and neutralizing antibody serology tests will be a priority going forward. There's a lot of interest relative to vaccines, as you might guess. So we are working closely with any party that, any developer that wants to advance development of these tests, and we look forward to more and more authorizations. It is a bit more complicated to semi-quant, a bit more complicated to do a neutralizing assay, or to correlate a given serology test to a neutralizing assay. So we look forward to working with you. And with that, that ends our introductory remarks today, and we can turn it over to questions. Thank you.
Operator will now take questions. Hello, operator, are you there?
Yes, I'm here. My apologies. We will now begin a Q&A session. To ask a question over the phone lines, please press star 1. Ensure your phone is unmuted and record your name at the prompt. Your name is required to introduce your question. To withdraw your question, press star 2. One moment, please, for incoming questions. Okay, we do have a few questions in queue. One moment, please. Our first question comes from Elisa Maldonado-Colbert. Your line is open.
Yes, thank you. Question regarding readers. After receiving an EUA approval for an antigen test with a reader, if we validate a new reader after our internal validation, do we need to submit a new EUA? Thank you.
That would probably be a supplement to your existing EUA. And the validation requirements would depend on how closely the second reader performs relative to the first reader. Without knowing those differences in how you do that, it's hard to predict and give you feedback in this short time we have on this call. But that would, if the antigen test itself is the same, just different readers, that will be more straightforward and can be a supplement to your existing application.
Thank you so much.
Once again, as a reminder, to ask a question from the phone lines, please press star 1, unmute your phone, and record your name at the prompt. Please limit to one question per caller. Our next question comes from Shannon Clark. Your line is open.
Hi, this is Shannon Clark with UserWise Consulting. We conduct human factors testing for medical products, and I did email you, Timothy, about this. For viral sample collection using swabs, the template includes 30 untrained subjects using the swabs. All are negative for SARS-CoV-2, and we directly observe the users to uncover use errors. My question is about the lateral flow test kit, because for those, it only requires 5 healthcare providers, 10 less than the FDA minimum number for human factors, no direct observation, and it requires 30 antibody-positive finger stick samples. So I was just wondering, is the purpose of this point-of-care testing to uncover use problems? And if so, why doesn't it include and require observation, and why does it require positive finger stick samples? But if the purpose of this testing is for clinical agreement or test accuracy, can you confirm that it is not considered human factors testing?
So there's a lot of questions in there. I'd like to unpack things one by one quickly. One is, why do we require 30 finger sticks? It's because that sample type is physiologically different than other serology sample types, and we want to observe performance, particularly on a lateral flow device, that is equivalent to serum plasma or venipuncture whole blood. And so we do need to see the full 30 positives. And that's a much lower level than we would normally require for an EUA, for a submission, and that's because this is EUA. There were a number of other questions, and I am not sure that I caught all of them. I think one of them is, why, for point of care, do we only require 4 or 5 or so users? It's because they are healthcare workers, and they are experienced in many cases with running point of care devices. So we don't feel like the users that have previous experience with various potential point of care devices, that we don't, under the pandemic, we don't feel like we need to, for EUA, we don't need to see quite as many as for non-healthcare worker situations where, because we require so much less of a validation relative to a non-EUA situation, we want to see more users who are unskilled and are not healthcare workers to make sure that they get accurate results. So there might have been some more questions in there. I just want to pause and see if there's any clarification.
Yeah, can you just confirm that the point of care testing that is required by the template is not human factors testing, and that you can just send a follow-up questionnaire to these healthcare providers? You're not required to observe for use errors as you would in traditional human factors testing?
So for healthcare workers in healthcare settings performing the point of care testing, we do not typically recommend for authorization for observation. Now, a developer may want to do that to make sure, that everything's working fine, that the workflow is great, that users don't have difficulty. For, more commercial marketing purposes.
Thank you so much. So we'll just proceed with advising our clients that the purpose of the point of care testing is for clinical agreement and or test accuracy, not for uncovering use errors.
Oh, that's true. I mean, performance in experienced hands, such as healthcare workers, sort of factors some of those things in already. Okay? All right, thank you.
Our next question comes from Dana Hummel. Your line is open.
Hi, thank you for taking my question. It's kind of a follow-up to the last question. For the section regarding point-of-care studies for the robustness of the test, testing different volume samples, different lighting, is that required to use finger stick whole blood samples, or could we use other sample types such as venous whole blood, plasma, or serum, or even controls like recombinant antibodies?
So I think you're talking about a serology test, and I do believe it does give you options on how to do that. Primarily, we want to understand how does the test perform if there are variables that aren't controlled by the test themselves, that are user-dependent, time, volume, observations. And even if you have like a smartphone reader, the lighting conditions matter. So for those, Toby, you might have this more at your fingertips than me, but those things can be tested more analytically in the lab rather than with actual finger sticks. And I'm just going to that section, but I don't think I have time to go to it right now. But any additional questions that aren't clear from the template of serology template for commercial manufacturers, please just ask our team. But I believe there is advice for how to do that without having to use actual direct patient material.
Okay. Can we perform these tests outside of the U.S., or are they required to be in the U.S.?
There's no requirement to perform those tests within the U.S.
Perfect. Thank you so much.
For those flex studies. Thanks.
Thank you.
Our next question comes from Neil Armstrong. Your line is open.
Thank you. Hi, Tim and Toby. Thanks as always for doing this and really helping us out. Our product is a general diagnostic device. It's not specifically to detect the COVID-19 disease, but because it detects changes in the body, we believe it's got an application in staging the progression or recovery from COVID-19. We got a lot of help early on in getting through the sort of template process and getting them into the right sort of format to put our information in. But recently we seem to have ground for a halt. We now believe we're in a position where we could put in a 510K for a general indication, and we appreciate you guys are just really busy on all the EUAs for the sort of normal testing. Should we put in a 510K for a general indication, knowing that people may be using it off-label for staging COVID-19 progression? Should we pursue the EUA route? Should we do both? If we put in a 510K, should we ask for an accelerated or expedited review? Really, what's the best way of us moving forward on something that probably isn't your first priority and may take a bit of review time?
So, yeah, things that are not targeting SARS-CoV-2 or antibody response to SARS-CoV-2, we have been bringing in non-microbiology review staff, even though there may be a microbiology component to it, right? Because of the desire to appropriately assess these technologies that can potentially assist in the care for COVID patients. So without knowing the details of the process and your specific situation, it's hard for me to advise. Where possible, we do want to follow the EUA route. We do follow closely the regs and guidance, obviously for determination of what qualifies for an EUA. And we've been, in some cases, in many cases, very flexible in adopting those things into the EUA process. For example, multi-virus or multi-analyte panel testing isn't something that was too clearly, something that's an EUA because it detects viruses and other respiratory pathogens that aren't directly COVID related. So if you've ground to the halt, and I hate to suggest this because my email box is absolutely crazy, but if you send it to me and you copy Toby, so send it to the template's email address, ask to have Toby and Tim, Dr. Stenzel, copied. I will get to it as soon as we can and help unblock this and make some specific suggestions for you to help you out.
Thank you very much. Really appreciate that. Great of you.
Our next question comes from Sarah. Your line is open.
Hi. We're looking for some clarification on the stance for university student testing. The updated FAQs indicate broad screening requires an EUA, but that healthcare ordered tests can be completed under the current authorized EUAs. If our student testing is completed under a healthcare provider order, as suggested was acceptable on our previous call from their high-risk status, do we still need to submit an EUA for this screening? And if so, can the data be retrospective or should it be collected prospectively?
So when it comes to molecular diagnostics tests and use in screening, if the test that you're using is an EUA authorized and or notified test, and it doesn't have any particular limitations, maybe due to sensitivity or some issues that we've seen, then they can be used in the screening / asymptomatic population when there is a healthcare worker prescription order. And we're allowing that very clearly on our FAQ site. FAQs, we encourage labs to accept these samples, even they may be screening or asymptomatic patients if they have a valid order, and to report those results. And for that purpose, there's no necessary change to an EUA unless that EUA wants to claim, that they have a certain level of performance in the screening population. And then we will want to see data in the screening population that would support that claim that their test has a certain level of performance in the asymptomatic or screening population. And our templates go into a lot of different details about, how to go about getting that. So, for example, if you already have an EUA authorized test, there's a pathway to add that screening asymptomatic claim. If you have an entirely new test and you want to go directly for that, there's a different pathway. If you want to have an OTC test, there's a different pathway, et cetera, et cetera. So there's the details have gotten such that it's hard for me to keep them all straight, but it's well laid out in the templates. If you have any questions about that, but if you're not going for, wanting to advertise that you're testing as a certain performance in the screening asymptomatic population, you're fine just accepting the healthcare worker order / prescription, doing the testing and reporting it. Hopefully that helps.
Yes, thank you so much.
Our next question comes from Martha Casasa. Your line is open.
Hi, thank you for taking my call. I am calling on behalf of the Clinical Lab Management Association. We are looking way in the future and wondering what is the plan once EUAs are lifted? What will be the status of all these assays and what will be required of laboratories to be compliant? Can you answer those questions, please?
Yeah, so I think what you're saying is once we begin to authorize regular submissions and potentially if the emergency should come to an end, what is the status of all these EUAs and what would be the plan for them at that point, correct?
Correct.
Yeah. So obviously this is very important. I will say that many of the previous emergencies are still declared, and most of them have not been converted to regular submissions. Some of them do, and we certainly wish them to be able to do that, and we work with them to achieve that, and we've achieved that, I think, for an Ebola test and a Zika test, for example. And so we don't know what decisions are going to be made regarding this emergency. I certainly don't foresee it coming to an end, but I'm not the one making the decision. And you can rest assured that we will have, a plan that will work for most everybody, taking all these considerations into place. So a lot of thinking has already gone into this. So the things that you say is as soon as we can make those plans public, we will do so. So we're already thinking about that. So I don't know that it should be a concern. I certainly don't personally think that it's a concern for any of the developers out there right now.
Excellent. Thank you so very much. Have a great day.
Thanks. You too.
Our next question comes from Pamela Turbeville. Your line is open.
We purchase qualitative serology tests, IgG, IgM, that already have an EUA. We read the results visually, positive or negative, and then our app digitizes the person's data, saves a picture of the cassette, and then reads the test results and sends a photo and the qualitative result to the person's phone. However, in parallel, we have a reader with an app that also quantifies the antibody scores from the serology test. We're not sharing the data with the individual, but we are using it to build validation data for an EUA application in the future. So our question is, can we proceed to test individuals, provide them qualitative data per the third party's IFU, and then capture the data, the validation data, and then compile it into an EUA application in parallel?
So just want to check a few things. So you are taking somebody else's test, probably, not your own.
Right.
And you've developed a smartphone app that takes a picture of the visually read lateral flow device. The data that's input, there might, and this potentially would go to the healthcare provider, medical record, and or a patient even. The test is visually read by the appropriate person who's authorized to do that.
Right.
In most cases, that would, at the moment, I don't think we have any point of care, but for serology, but which we are very interested in, and we'll work with developers to achieve that. But they're in the moderately into high complexity lab situations now. So those workers would read that test. They would record that result in your software package, along with a photo capture of the results. Is your device currently reading the strip itself and making a determination of either qualitative results and or semi-quant results?
Okay. The strip itself provides the qualitative results, so we tell the person right away yes or no. So that is just an operating procedure. That's done. But what we're doing in addition is we take the test cassette, we're putting it into our device that sits on a smartphone, insert it, press a button, and it quantifies it. We record that data. We're using that data for our own future submission for EUA.
Yeah. Yeah, so that would be considered a device because you're actually doing analysis of the results of the test and coming up with a new output, right, based on that device's analysis. So there are two pathways. There's an easier pathway and there's probably a very difficult pathway. One is to work with a given developer who already has a test, and you do the performance measurements and validation on that test for use with that test. And that could be, there's various pathways for that. If you're, the test developer themselves can submit everything, and update their EUA potentially, or you can stay independent. We can figure out how to do that. Basically, exchange write-up reference letters. And we would probably update both, give your smartphone application its own EUA, and then we would also update the package insert of the test to say that it can be used for this device. If you wanted to have a more broad claim that would work on multiple platforms, multiple tests, it would be a little bit more challenging because there's obviously variability in those tests. And that would be fairly challenging given, sometimes the reads are different, locations are different, colors are different, intensities are different, et cetera, et cetera, et cetera. Okay?
Got it. Thank you so much.
Our next question comes from Gabe Oland. Your line is open.
Hi, Tim. Hi, Toby. Thank you for everything that you and others are doing. I'm working with a university-based lab that's developing a next-gen sequencing platform for massive diagnosis for students who are coming back to campus on the order of about 100,000 tests in a single day using saliva. In the molecular diagnostics template, there's a requirement for alternative respiratory specimens to reach 95% PPA. With an NP swab sample. And I'm just wondering, knowing that specimens in different areas in the respiratory tract have different concentrations of virus, if you think that this is going to be a pretty rigid rule to maintain 95% PPA in this kind of a setting.
Also, are you going to try to pool actual saliva samples before you begin testing, or you're going to just do a massively parallel and test saliva samples individually and use barcodes to convolute and deconvolute?
The latter, so massively parallel, and they'd be unpurified saliva specimens, which is what allows it to be so scalable.
Okay. We've found saliva to be an incredibly unpredictable and challenging substrate. I mean, we've really not ever considered testing saliva for a respiratory virus and authorized it before, to my knowledge. This is incredibly unique, and we're open and flexible and adaptable to different approaches. But we have seen and denied some saliva submissions because of performance. And we are aware of multiple developers who have just decided that saliva is too tough. Obviously, there's advantages to saliva, right? Easy to collect. You don't need, you may not need transport media, or if you do, it's in a device that's easily used, and we've authorized tests with some devices already. And you don't need, swabs. You don't need VTM per se, and so there's huge advantages to being able to use saliva. So we just ask that developers, and if they want to, and saliva can be added to an EUA test without an FDA submission if it's an LDT. Obviously, kit manufacturers, it's a notified pathway. They can validate saliva, notify the FDA, and in 15 business days submit their saliva EUA package. But for labs that want to either alter a kit or alter their own LDT, we don't require an EUA submission for that. But we do ask that you validate it well. And we have come to believe that comparison to NP swab is critical to understanding the performance and the performance limitations of saliva. I don't think we require that much. I mean, it's basically 30 positives. And we understand that nasopharyngeal swabs are sometimes hard to obtain. A good second choice for an NP to saliva comparison would be a mid-terminate sample, mid-terminate swab sample. I think most students, patients would much prefer to have a mid-terminate to a nasopharyngeal swab. So that is also acceptable to us right now. We are trying to discourage nasal swab to saliva because there is the potential for additional loss of sensitivity in a nasal swab. And we have found in many cases now that comparison is not the best comparison to use going forward. So hopefully that helps.
Gotcha. It does, yeah. I think, we have a pretty strong limit of detection with our assay, and so I think maybe some of the loss in sensitivity can be made up for by frequent testing on a more regular basis. So just curious if that would come into consideration when comparing to the NP swab. But great. Thank you.
We do allow performance of a molecular test and also direct antigen test down to an 80% sensitivity or PPA for almost anything. We do request that anything that generally falls below a 95% sensitivity compared to a good swab sample or another good test be labeled with the negatives being presumed negative rather than actually negative. Because if you're missing up to 20% of positives, then we think that clinicians should know that and make sure that the situation that they're getting that result on doesn't warrant a follow-up swab test, for example, to make sure the patient is absolutely negative.
Okay. That's good to know. That's actually what we're intending, is to then, if they get a negative, they would then get an NP swab. So thank you very much, Tim. I appreciate it.
Uh-huh.
Our next question comes from Daniel Marcus. Your line is open.
Hey, so my question is related to the notification pathway. Specifically in the May 11th guidance, you guys use the term distribution and that it's okay for commercial manufacturers to distribute the tests. And in this conversation, you mentioned the term market. I'm just wondering what that looks like with respect to commercialization and what developers and manufacturers are capable of doing as far as being able to seek any sort of financial reimbursement for their tests. That's question one. And question two is you guys specify basically two entities where these tests can be distributed, which is high complexity CLIA labs and healthcare workers at the point of care. So I'm wondering what that definition of healthcare work at the point of care looks like, and whether those have to be high complexity CLIA lab personnel, or they could be any healthcare worker at the point of care. But I'd love to get the first question answered first, and the second one's just icing on the cake.
I'm gonna actually reverse them, because I'm gonna hand the first one over to Toby for her thoughts after I first try to address it, just to make sure she doesn't have anything else or any corrections on what I say. But
sure, I'm happy to.
We authorize tests under EUA, and we give a clear indication of whether that test, where it should be performed, whether it's a high complexity lab, moderate complexity lab, point of care, which are healthcare settings with non-laboratory personnel, non-lab collection, including home collection, non-lab testing, including home testing, and whether that non-lab testing is by prescription or it's over the counter without prescription. So there are all these various scenarios. And if we have said that either a test is moderately complex or highly complex, then it should not be used in the point of care setting, except as is allowed under a moderately or high complex lab's CLIA certificate. So it cannot be performed only under a waived certificate. Hopefully that provides some clarification.
Okay, that's within the context of the EUA, but I'm asking vis-a-vis the notification pathway. What's acceptable in addition to in terms of distribution of the tests, in addition to high complexity CLIA labs?
So,
and
so
let me, Toby, let me.
Oh, go ahead, Tim.
Yeah, let me start that question. All right, that answer. So under notification, the test can be performed in a high complexity lab. For those entities that are labs and have an LDT and they want to have their notification posted, we'll post it on the FDA website. All kit manufacturers via the notification pathway do go on that list. It is my understanding that, but we would have to refer you to CMS on reimbursement, that tests that show up on the notification list but perhaps aren't yet EUA authorized can still be reimbursed. And with that, I turn it over to Toby for filling in more details and or correcting me.
No, thanks. So under notification, as noted in the guidance, there are all of the tests that are being offered without or prior to an EUA default to high complexity, and that's under the CLIA regulations. So because they're high complexity, the only way that they can be used at the point of care is if that point of care site is covered by the laboratory's high complexity, or by a high complexity laboratory's CLIA certificate. Sorry about that. So I think that gets to your second question of what the setting is where these tests can be performed while they're being offered under this policy. Is that correct? Is that what you were asking there?
That is what I'm asking. That does clear it up. One sub question of that, not to hog the mic, is so you guys are allowing, or maybe this is more of a CMS question or a CLIA question. You guys are allowing high complexity CLIA labs to do, I guess, off-site testing outside of the auspices of their own campus. So if they wanted, if a high complexity lab wanted to be able to do testing at a school, or is it just totally outside of your domain?
That would be a CMS question. But what we're generally referring to here in the guidance is situations where and this happens outside of the public health emergency, there may be situations like in a hospital where the hospital's laboratory does some of its testing at a near patient site, so at the bedside. But it would still fall under the laboratory's certificate.
Okay.
And then for your reimbursement question, that really would be a CMS question.
Okay, so I mean, it's not necessarily reimbursement, more so than, you know, is it possible for a commercial manufacturer to just outright sell a test through the notification pathway, or is that a CMS question?
Oh, yeah, the notification policy applies to commercial manufacturers distributing their test kits for testing, and that is intended to mean sale of your test. We're not saying that you should distribute them without.
Okay, that's what I wanted to get clarification on. So you guys have answered my questions beautifully. I will stop hogging the mic. I appreciate your time.
Operator, do we have any other questions in the queue?
We'll take our next question.
Is our operator still there? Do we have another question?
Victor, are you on the line?
Sorry for the technical difficulties on today's call, everyone. Thanks for your patience.
Victor is
there
on
we go.
the line.
My apologies. Yeah, my mic was being odd. I apologize. We do have a question in queue, and that question is from Nirmal Ravi. Nirmal, your line is now open.
Thank you. Thank you for these really informative sessions. I work with a clinical diagnostic laboratory that's outside the U.S. that's using a U.S. FDA EUA test, PCR test for COVID-19. So my question is the FDA accepting any user data regarding test performance? Because we are seeing between 10 and 20% invalid test results. So would the FDA be happy to receive those data? And if so, how can I do that? Thank you.
Yeah, I'll start off. Toby may have something additional. So I think what you're talking about is if you're seeing subpar performance or issues with the test, even though you might be using it outside the U.S., are you allowed to report that to the U.S. FDA? And yes, absolutely. You can report that through the MedWatch program on our FAQ page. You can also email our template email address. You can also report that to the company. We do require companies to record all complaints, do a root cause determination, and find out if it's verified, and in situations that require it, reporting those results to the U.S. FDA, even though they might occur outside the U.S., as long as that test is also marketed in the U.S. and could impact U.S. healthcare. Toby, is that pretty good?
Yeah, that sounds right to me.
Thank you very much. And which template email address should I send it to?
It's CDRH-EUA-template. And it should be on, at fda.hhs.gov. And it should be on the slides that are being displayed with the presentation today.
Thank you both very much.
Our next question comes from Lubna Syed. Your line is open.
Hi, Tim. Hi, Toby. Quick questions for you about the non-lab use template as we are running out of time here. First question is regarding the exclusion of individuals who have experience with diagnostic home use tests like glucose monitoring tests. I'm assuming that applies only if your test is a finger prick type of non-lab use test as opposed to nasal swab. Is that correct?
No. In general, we want to assess untrained patients and somebody who has routinely been performing testing on themselves at home will have an advantage over an untrained patient or consumer. And we're really trying to understand on the small number of samples that we're requiring in an EUA in that setting, that we really understand the performance in a totally untrained user, because that's the vast majority of potential users out there would be somebody who's never performed something like this before.
Okay, thanks. And then just really quickly, I'm wondering in terms of the translation to Spanish of the quick reference information, if the software user interface is not translated, but that's the quick reference information is utilized in the studies, if that is going to be acceptable for OTC claim.
I'm not sure I understand your question. You're talking about what language it has to be in, because and what population would be needed and comprehensive.
Yeah, I think there's a strong requirement to include your quick reference information in both English and Spanish for over-the-counter claim, is what the template suggests. And I'm asking if the quick reference information is translated, but the graphical user interface of the app, the step-by-step steps are still in English, and those are the tools that are utilized as part of the study. If that will be adequate in terms of translation, that you don't need to also translate your graphical user interface in order to obtain that over-the-counter.
Into Spanish, yeah.
Yeah.
Yeah, bare minimum, we want to see things in English for the U.S. population, but to the extent you want to include language for other speakers within the U.S. And the populations, then we salute that. But we want to see, at a minimum, a performance with English language in the instructions and in the app. And it's up to you whether you want to go beyond that.
Okay, and then I do have others, but I'm going to refrain from hugging the mic. I'll turn it over. Thank you so much.
Our next question comes from Kodumudi Venkat. Your line is open.
Good afternoon. Can you hear me?
Yes.
For taking the question, my question is, you have so many priority UEA things have come for serology. Now the semi-quantitative and quantitative tests have come. My question is, you have so many pending qualitative serology tests. So is there any timeline when the backlogs will be cleared? Are you getting so many additional support so that these pending qualitative serology tests can be looked into and then the decision will be given?
We're always looking at adding staff, and we are hiring. And we are looking at ways to reduce the time it takes for us to evaluate. And we do appreciate those developers who work closely with us, and as is our desire to see NCI testing to work with us if that's what we ask. And we are working really, really hard. That's why we're so appreciative of the opportunity to have the notification pathway, so that developers, once notified, can sell, market, distribute their tests in the U.S. So, but getting to that EUA decision is also very important.
Actually, we have submitted our test to NCA 2 months ago. We haven't heard anything from them yet. I think they are busy. And also, main issue with us is it is in the notification list, but many labs, they insist it has to be a UEA approved authorized test for them to procure. So they are interested. They did their independent validation, but still they are insisting on a UEA authorization for them to procure. So is there any way you can help?
So we would ask that NCI is working very, very hard. They do have a number of tests to test, and they're working very hard and as fast as I think they can. And so, you should have a contact at the FDA who can, at least on a weekly basis, give you an update on the status of things, and I would have you direct your questions to that contact. And again, there's a notified pathway. We understand what you're saying. We're doing the very best we can here. All right.
Thank you.
Next question.
Our final question comes from Steve Skaggs. Your line is open.
Hi, thank you for taking my call. I just have a question about the fact sheet that is supposed to accompany the kits. Is there a template available? I haven't seen anything, or is it just general information from the IFU that we put together for a more layman explanation of how to use the kit?
Yeah, so we don't typically ask developers to weigh in on those fact sheets. They're pretty standard given a specific technology. They have undergone some revision over time to be more helpful, but that's something that the FDA drafts and provides during the process, and then posts with each authorization.
Great. Thank you for your help. I appreciate that.
You're welcome. You too.
Now I'll now turn the call over to Irene here for closing remarks.
Thank you. This is Irene here. We appreciate your participation and thoughtful questions. Today's presentation and transcript will be made available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Tuesday, August 11th. If you have additional questions about today's presentation, please email cdrh-EUA-templates@fda.hhs.gov. As always, we appreciate your feedback. Following the conclusion of today's presentation, please complete a short 13-question survey about your FDA CDRH virtual town hall experience. The survey can be found at www.fda.gov/CDRH webinar immediately following the conclusion of today's live webinar. Again, thank you for participating. This concludes today's discussion.
Thank you for your participation in today's conference. You may now disconnect.